What is up, guys? I'm back with another video, and I'm really excited about this one because this is a mnemonic that I have worked on for about a week now to help me remember all of the steps and in their particular order for the urea cycle. So I want to remind you that the urea cycle, the prominent reason, this is to put it as simple as needed, the prominent reason of the urea cycle is when you have a basically excess protein in the body. Excess protein, or say you're having to break down protein for energy, or whatever the case may be, you basically, when you break down the protein, and remember proteins are made of amino acids, chains of amino acids. When you break down proteins and when you break down those amino acids, you form a toxic byproduct. And in high concentration, it's extremely toxic to the body. And this is called ammonia. And the chemical structure for ammonia is NH3, so 1 nitrogen and 3 hydrogen. When this builds up in the body, this is extremely toxic and can cause encephalopathy, a lot of neural symptoms, then which will result in neuromuscular symptoms, and it can be life-threatening. So we have to do something about this ammonia. And what the urea cycle does is it converts the ammonia, remember it's NH3, into something called urea. Why that's important is because ammonia can't be excreted from the body effectively. It can be converted to kind of other more non-toxic forms, but there's a limit to that. So we need to convert this ammonia into something called urea. And urea can easily be excreted by the kidneys so that we aren't harmed from the toxic buildup of ammonia or NH3. So that's basically what the urea cycle does. But it doesn't end there. There's a lot of complex steps that are extremely hard to remember for the urea cycle. And this is extremely high yield in the biochemistry section for a lot of medical board exams. And there's various defects in these enzymes that can present when you're in an infant. And so you need to know this pathway and also the enzymes that basically are for the different parts of the pathway to be able to get these questions right. Now, you may have looked at tons of diagrams for this pathway and thought to yourself, how the heck can I remember this? How am I going to remember all the enzymes, where they go, what they do, all of these things? Well, I have a really clever mnemonic to help you with that. So let's get right into this. I want to start off by saying that First Aid has a mnemonic for this pathway. And I read the mnemonic and I was entirely lost. I was like, I don't think I'll ever be able to remember this mnemonic, considering there's like 200 or 300 mnemonics all throughout First Aid. And so the mnemonic is this. First Aid gives a mnemonic, says, ordinarily careless crappers are also frivolous about urination. The first problem I find with this mnemonic is that that entire mnemonic only gives you the first letter for each of the supposed substrate and products that go throughout the urea cycle. That's not going to help when you have parts of the urea cycle, for example, Arginino succinate is one of the substrates or products and then turns into a substrate in this pathway. Then that goes into arginine. Do you see why that mnemonic would be a problem in using this for the urea cycle? Because both of them start with the letter A. So you could easily mix up Arginino succinate arginine and get it in question entirely wrong if they ask about an enzyme that happens here or an enzyme that happens prior to here. And they want to know, does this go up? Does this go down? Does this go up? Does this go down? And you'll miss the entire question just because the mnemonic you may not have remembered. The mnemonic was not enough to separate some of these kind of hard parts of the pathway. So I am going to give you a mnemonic that helps with this entire pathway. Let me write it down at the bottom right now, and I'm going to show you how easy it is to use this. This is all you have to remember. All you need to remember for this pathway: old car seats are so large. That's it. That's all you have to remember. Old car seats are so large. That is far easier than the mnemonic that's in First Aid. On top of it, my mnemonic that I've pretty much developed and thought about will give you a lot of time, in not all of them, but in a lot of these, it'll give you more than just one letter or more than just one hint of some of the substrates, and you'll see that as we go forward. So let's get right into this pathway. I told you that the point of this pathway is to break down proteins, and remember, these are just polymers of amino acids, and you're going to break down these proteins. Say you have excess, or you need it for energy, and you're going to get ammonia. And I said that you cannot have too much ammonia because it's toxic in the body. So this ammonia is going to combine with CO2. CO2 is pretty much all up in the body because we use oxygen that we breathe in to go through aerobic respiration and form ATP, and then CO2 is a product of that. So we have to breathe that CO2 out, but we can also use it in this pathway. So ammonia plus CO2 goes to form something called carbamoyl phosphate. Carbamoyl phosphate. Now, let me begin to show you, in this next step, I'm going to show you where the mnemonic comes into play. But first you need to know the enzyme that does this. It's pretty straightforward, except for one little point. The enzyme that does this is called CPS1. Carbamoyl phosphate synthetase 1, or synthase 1. So that's really easy. Obviously, you're making carbamoyl phosphate, so the enzyme is called carbamoyl phosphate synthase or synthetase. It's really not important. But then here's the difficult part. There's a Roman numeral 1 right here. I want to recall, if you've studied in nucleotide production or synthesis pathways, that carbamoyl phosphate is involved in nucleotide synthesis. And carbamoyl phosphate is also involved here. And there's a step in this where this is converted to something else, but the enzyme that does this is called carbamoyl phosphate synthetase as well. But this is different. This is carbamoyl phosphate synthetase 2. So here's the first thing. How do you remember which one is 1 and which one is 2? Because believe me, I've seen QBank questions that will do this to you. They'll try to get you to put Roman numeral 2 for the urea cycle or Roman numeral 1 for the nucleotide synthesis pathway. And so you will miss a question if they do that. How do you remember it? When you look at the Roman numeral 1, it looks like the letter I, a capital letter I. So I think of I for inside. The reason I do that is because purine synthesis does not happen in the mitochondria. But guess what does? The urea cycle, the first two parts that I'm teaching you right here, happens inside an intracellular organelle. I for inside. So that'll help you remember it has to be a Roman numeral 1. Okay? So we've gone through the first step. Ammonia is combined with CO2 to form carbamoyl phosphate using the CPS Roman numeral 1, that's very important, enzyme. Now, carbamoyl phosphate is going to combine with something, and it's going to combine with the last byproduct of this entire urea cycle, and that thing is called ornithine. Ornithine. And it's going to combine with the carbamoyl phosphate that was produced in the kind of this pre-step. Sometimes it's not necessarily even part of the urea cycle, you could say, but you need to have that carbamoyl phosphate to go through this. So ornithine combines with carbamoyl phosphate, and it's going to form something called citrulline. Now we can begin to see where the mnemonic's going to come into play. Check this out. The O stands for ornithine. Well, you may say, well, that's only one letter. How is that going to help me? Well, look at the entire pathway. I know I haven't drawn it all yet, but if you look at a picture of the entire pathway, citrulline, Arginino, succinate, arginase, all these different parts, nothing else starts with the letter O. So you only need one letter here. There's no confusion on that. O is for ornithine. So that's why it comes first. In this mnemonic, old car seats are so large, O for ornithine has to combine with what? Carbamoyl phosphate. And look at the beginning of carbamoyl. Car. There it is. Car. That gives you 3 letters to... Let me zoom in here. Hold on. So that gives you 3 letters. Pretty much everything you need to remember, oh, this C is for carbamoyl phosphate, not for the other C, citrulline. Do you see how you can mix that up? That's why it's so important to have a mnemonic that has more than just one letter. So, old car seat, so this tells you ornithine, and then ornithine is going to combine with carbamoyl phosphate, and it's going to form what? Citrulline. Now you'll say, how does seats help me with citrulline? Just pronounce seats, and does it not sound like citrulline, citrulline, citrulline? So that is far more helpful than putting another C in this mnemonic, and then getting the two really important first, second, and third steps mixed up right off the bat, and you missed the question. So seats for citrulline, okay? Basically pronounce it like a seat. So that covers that. So we have ornithine, carbamoyl phosphate, and we have citrulline. Now the next thing, what's going to happen? This citrulline is now going to exit the mitochondria. So that's another important note I want to emphasize. All of the steps I've just said, the production of carbamoyl phosphate, the production of citrulline with the combination of ornithine and carbamoyl phosphate, all of these 2 steps happen in the mitochondria. That is so high yield because they will ask you referring to the urea cycle, they'll give you a bunch of enzymes. They'll say which of these enzymes are located where, and they'll say, is it located, and they'll give you all these spots. You need to know that the first two steps are located in the mitochondria, so therefore those enzymes need to be in the mitochondria, or at least get there, right? So those are the kind of questions they can ask you. So the citrulline leaves out. We have the citrulline that is produced and it leaves out. Now we're in the cytosol. And I also want to say this entire process or pathway, the urea cycle, although we have these first two steps over here that's going to take place in the mitochondria, and all the rest of the steps are taking place in the cytosol, the entire process predominantly takes place in the liver. Very little bit takes place in the kidney, but you just need to know liver. It takes place in liver hepatocytes. But then when you go into the liver, there's various parts of liver cells, mitochondria, cytosol, so that's what you need to know. The urea cycle takes place in the liver, but the first two steps takes place in the mitochondria, the rest is in the cytosol. Okay, so I got past some of those sloppy details. So we have our citrulline that we've produced. This citrulline is now going to go into, and let's look at our mnemonic to help us, old car seats, ARSO. Say it together, ARSO. Argino succinate. R for argino, succinate for the succinate, so that we know that combines together. Argentino succinate. That is the next product that citrulline is going to be converted into, but it's not going to do it by itself. Notice that this citrulline has something called Arginino at the beginning. That's because there's a structure very similar to arginine that is within this molecule now. Arginine cannot just be produced out of thin air. There needs something to be combined with it. And something is going to come into the pathway right here. And what's going to come in is called aspartate. You may have heard of it. It's an amino acid. Aspartate is going to come in and combine to form Arginino succinate. This is another point I want to really stress to you. I have gone through questions where I've used some mnemonics that I didn't think were very helpful, and I had, a mnemonic that only gave the first letter for say, and then they give you something they'll ask you some question relating to this aspartate, and then eventually this Arginino succinate goes in through another process and it turns into arginine. So here's what they'll do. They'll try to get you to think that aspartate is here, and then arginine is up here. So how do we remember that? Well, this mnemonic helps you with that. After the old car seats part in the mnemonic, do you see ARSO? Aspartate does not fit into the word R. AR is for arginine, it doesn't have an AS in there. It's ARE for R for arginine. So that helps you remember that as far as what's in the actual pathway, what's being produced for our mnemonic, arginine is what's going to be all within the pathway. And then the thing that's coming from the outside is the aspartate. So that's another thing that can help you. Now, let's go back to the enzymes that are involved. Citrulline combines with this aspartate to form Arginino succinate. Watch how easy this is. You really don't even need to memorize this. Arginino succinate synthase. It's that simple. Literally, you don't have to memorize it because citrulline combines with aspartate to form Arginino succinate. The name of the enzyme is Arginino succinate synthase. We are producing, or like synthesis stands for production of, we're producing Arginino succinate. So that's straightforward. Also, another thing to keep in mind, if you remember, anytime you see synthase, almost all the time, not all the time, but almost all the time, ATP is needed. So ATP is needed in this pathway. When you see synthase, think, oh, I need ATP in this pathway, for most cases. So we have this aspartate and ATP is needed to do this reaction. We're turning citrulline into Arginino succinate. We can remember citrulline because of the seats in our mnemonic. And then the very next two words, ARSO, Arginino for R, and then succinate, this is the so in the mnemonic. So just kind of say those together quickly. ARSO, Arginino succinate. Succinate for the so. Okay, so now we have our Arginino succinate, but we need to remember now that Arginino succinate goes to arginine. Well, it's kind of easy to do that because Arginino sounds like arginine. So it would make sense that we're going to now convert this to arginine. But if you can't remember that, the large should help you. ARG for arginine, right? That's straightforward. Arginino succinate, ARG in large for arginine. Now you may say, well, what about the AR in ARSO, the ARSO part, the AR and R? Well, why wouldn't I mix those up? Because that's what the so is for. Arginino so, S starts, right? S for succinate. So that helps you remember, oh, this is not arginine right here. the arginine is right here because the so after the R helps you remember that this is the bigger word to start with. Okay, so we've converted the arginino succinate to arginine. What's the enzyme that does this? So it goes by two names. Arginino succinate, there it is again, so that's pretty easy. It's literally the reaction of this whole thing. It's the substrate of this reaction. Arginino succinate lyase is one name you could see for this. Another name you could see for this is just arginino succinase. Arginino succinase. So either one is correct, but basically it's pretty easy because arginino succinate is right here, and naturally you are breaking it down into arginine, okay? And our mnemonic can help us remember what it breaks down into. All right, so now we have arginine, and you know what? Now there's only one more step, and we already have talked about ornithine. I said that ornithine was the end product at the beginning of this video for the entire urea cycle. And then so that we don't have tons of buildup of ornithine and nothing to do with it in the body, we will utilize ornithine again to combine with carbamoyl phosphate, back right up at this step, and it's going to combine with carbamoyl phosphate to form citrulline, or citrulline as I like to say it to help with the mnemonic. Now, in this step, arginine going to ornithine, it releases something, and it's the most important part of the whole pathway. Arginine going to ornithine releases urea. Okay? Arginine going to ornithine releases urea. And urea was the whole thing we were trying to form the entire time. We were trying to take this toxic byproduct up here of ammonia that if it builds up in the body can be fatal to your health, and we're trying to convert it to something that the kidneys can excrete. That thing that the kidneys can excrete is, so the kidneys excrete this, it's called urea. Now, the enzyme that does this, and again, it's so simple you really don't even have to worry about it, arginase. That makes sense. Arginine, arginase. That would logically make sense that that enzyme would go with arginine and then break down, and then that, you know, since that's the final step, naturally that's when urea would come out. And then we form that ornithine, which I said was the final byproduct. One last thing I didn't mention is in the step from arginino succinate to arginine, you will release something called fumarate. Do you remember where fumarate comes from? Fumarate is involved in the Krebs cycle. So by breaking down these proteins, you can send fumarate into the Krebs cycle, and that can help aid in the process of the Krebs cycle, or what's also called the citric acid cycle, to form all of the NADH and FADH2 and whatnot. Okay? All right, so that is pretty much the pathway for the urea cycle. Now, we have the whole pathway down, right? We have the pathway. I'm going to erase everything to make this more simple because we've talked about it. I'm going to quickly just write the substrates and products just so I can talk about some of the diseases that come into play. So we have ammonia that has come from the breakdown of proteins. And we're going to combine that with CO2, which is all over the body because we're constantly using oxygen to make energy in the form of ATP. So we have the CO2 that we need to get rid of anyway through the lungs and through this pathway. It's going to combine to form carbamoyl phosphate. And that carbamoyl phosphate is going to combine with ornithine coming from the final byproduct of this entire pathway. It's going to combine with the carbamoyl phosphate to form citrulline. All of this takes place in the mitochondria. That's very important to know. Those first two steps take place in the mitochondria. We are now going to move the citrulline outside of the mitochondria into the cytosol of the hepatocytes. So now that we have the citrulline in the cytosol, the citrulline is going to combine with aspartate to form arginino succinate. The arginino succinate, and remember this step takes ATP because the enzyme that does this is called, do you remember what it's called? Arginino succinate synthetase or synthase. And you remember anything that has a synthase for the enzyme, the majority of enzymes, not every single one of them, I know there's a few exceptions that we learn about, but the majority of enzymes that have synthase at the end requires ATP. So we're going to put the aspartate and we're going to use ATP for energy, and we're going to turn citrulline into arginino succinate. Arginino succinate is going to be converted into arginine. That's pretty easy to remember. Arginino, it's literally in the word, arginine. Now the next step, and remember what that's going to release? That's going to release fumarate. Fumarate. Sorry if I misspelled that. Fumarate. And fumarate is a reactant in the Krebs cycle, so that can help us to form FADH2. So then arginine is now going to be converted into ornithine. And remember, ornithine, as I said, is the final byproduct of this whole cycle. When it forms that, it's going to release urea. And that's the whole thing that we're trying to do. We're trying to convert this toxic byproduct up here of ammonia into urea. Now, here's where it gets interesting. The enzymes throughout this whole process, and I'm going to go ahead and write them in right now because I think there's one of them I didn't label. And I'm going to show you a tip to kind of remember it. The first enzyme is carbamoyl phosphate synthetase 1. How do we remember that? That it's Roman numeral 1 and not Roman numeral 2 that's involved in nucleotide synthesis, because the Roman numeral 1 looks like the letter I, so think of inside, and this is happening inside mitochondrial of the hepatocytes. So that's the first one. Then carbamoyl phosphate combines with what? With ornithine to make citrulline. Now, I didn't mention this earlier. Ornithine transcarbamylase is the enzyme that does that step, that will form citrulline from ornithine and carbamoyl phosphate. This is very high yield because there's an inherited deficiency that babies can present with, inherited deficiency, that's called ornithine transcarbamylase deficiency. In other words, if you don't have this enzyme working, you are going to, first of all, have a buildup of ammonia. So the baby will have really high levels of ammonia. They're also going to have a buildup of carbamoyl phosphate, because I want you to notice that not only are you backing up the ammonia in this process, CO2 doesn't, you can have some change in CO2, but you can also just breathe that out, so that's not really a problem here. The ammonia builds up, but the carbamoyl phosphate builds up. So they're going to give you a question. They're going to describe a baby who has hyperammonemia, right? High levels of ammonia. And then they're going to tell you that this baby has been found after genetic testing to find a defect in ornithine transcarbamylase. What are the changes in all these substrates? What's going to go up? What's going to go down? And they'll give you like a little chart. And in one of the lines it'll be like, this will be citrulline, this will be carbamoyl phosphate, this will be NH3, and then this will be whatever. We'll just say X. And they're going to say, will it go up? Will it go down? For each one of these things. In ornithine transcarbamylase, if this is not working, if this doesn't work, naturally, you can no longer get to citrulline. So citrulline will go down in this disease. What about carbamoyl phosphate? We can still make carbamoyl phosphate because CPS1 is working. We just don't have an ornithine transcarbamylase enzyme working. So that means carbamoyl phosphate is literally building up in massive quantities right here because it can't continue on to be converted into citrulline. So carbamoyl phosphate will go up. The ammonia levels will naturally go up because as carbamoyl phosphate builds up, the pathway is being blocked up. So ammonia is being built up because you have too much carbamoyl phosphate. So now the ammonia and the CO2 can eventually, you just can't convert to that much to carbamoyl phosphate anymore. So the ammonia will build up. So all of that's important to know. So a disease called ornithine transcarbamylase deficiency is important to know for the STEP exam or for whatever board exam you're taking. Another disease that they're going to try to get you with. There's another enzyme. And the enzyme, remember, that's really high yield at least. Citrulline being converted to Arginino succinate. That enzyme, remember what I said it was called? Arginino succinate synthase. Now, imagine, let's take the same table over here that we just went through. Right? We just went through this table. Now we had citrulline, carbamoyl phosphate, NH3, but now we're going to add in, let's say, we're going to add in Argino. I'm just going to put Argino like that. Arginino that stands for Arginino succinate. So now, if you had a deficiency in this one, say this one wasn't working, now what's the problem? Well, CPS1 is still working. Ornithine transcarbamylase is still working. So all of these substrates are not going to necessarily, they can keep going. But here's where the problem is. The problem is when you reach citrulline, now you've reached citrulline, and you cannot continue further to get any of these other downstream products. So what does that mean for the level of arginosuccinate? Will it go up or will it go down if this doesn't work? It's going to naturally go down. So argininosuccinate goes down. What's going to happen to the level of citrulline if this enzyme right here isn't working? The level of citrulline can't go through to be converted to argininosuccinate. So the level is going to go sky high. So citrulline will go up. What's going to happen to, let's go back to carbamoyl phosphate. What's going to happen to carbamoyl phosphate? Well, carbamoyl phosphate, even though ornithine transcarbamylase is still working, eventually there's just such a high backup of citrulline that it's going to begin to feed backwards, and you're going to have a buildup of carbamoyl phosphate. That's going to go up because the whole pathway is being blocked right here. So everything in back of it is going to be increased. And then naturally ammonia goes up. So ammonia will go up. And that's the real dangerous part of this whole thing. The fact the ammonia goes up in these diseases is what's going to what can cause hepatic encephalopathy in babies and even kill the baby if it isn't dealt with. Okay, so those are two really high yield diseases. Ornithine transcarbamylase, and I'm going to write it underneath the ornithine transcarbamylase deficiency down here. The other one is Arginino succinate synthase deficiency. They're very high yield. Now, I'm only talking about those two, but just you could see that any of these other ones now is pretty straightforward. You could now figure out what's going to go up, what's going to go down. For example, what if this enzyme was messed up right here? What if Arginino succinase, it's also called lyase, remember? It can also be called lyase. What if that didn't work? Well, if that one didn't work, now in that little table for this inherited enzyme deficiency, in this situation, what would happen to citrulline? It would go up, right? Because it's backing up everything prior to that. So citrulline would go up. What would happen to carbamoyl phosphate? It would go up. What would happen to NH3? It would go up. And what would happen to arginine? What if I ask you that? What would happen to the arginine levels? It would go down because you can't convert it to that, okay? And then the Argentino succinate, I didn't add this in here, but if there was a line for Argentino succinase, that would go up as well because it's being blocked from going to be converted into arginine, okay? So I hope I wasn't talking too fast. I showed you the mnemonic at the beginning to remember this whole cycle. And let's just write the mnemonic one last time with nothing on the page and then talk through the mnemonic so you have a final understanding. The mnemonic is old car seats are so, and remember I say that really quick deliberately because that's kind of one thing, are so large. The old is for ornithine. The car is for carbamoyl phosphate. The seats, remember pronounce seats as citrulline. The arso is for Arginino succinase. And then the large, you look at the ARG, that is for arginine. Okay? That has helped me so much with those questions and not missing them anymore. I got tired of understanding the whole pathway and kind of having it in my short-term memory, but then forgetting it like a month later. Drill this mnemonic through your head. Just keep writing it down and saying it to yourself and remembering what stands for what, all of the different parts. For example, don't get mixed up and be looking for something with an S. Pronounce seats as for citrulline. Okay? I hope this helped you. If it helped you in any sort of way, please like, subscribe, share the video, and I will see you in another video. Bye, guys.